Thanks. We're going to get started here. Welcome back, everyone. This is session 3, part 1 of a 2-part session on deposition and effects of mercury and toxics. I'm David Schmeltz with EPA, and this is my co-chair.
Colin Eaglesmith with USGS.
Thank you, Colin. And we're just going to get right into it here with our first speaker, Ting Wang from UMass Lowell.
Thanks for the introduction. Hello, everyone. I'm Ting Wang. I'm working this project when I was postdoc in UMass Lowell. And thanks all of these co-authors' help, and without them, I cannot finish it. And today I'm going to talk about atmosphere surface exchange of gaseous elemental mercury in a salt marsh estuary in Massachusetts. The reason why we want to do this study, because the terrestrial ecosystem has been shown as an important global atmospheric mercury sinks, which is driven by plant uptake of atmospheric gaseous elemental mercury. Well, the study mostly in the upland forest ecosystems, but the importance of GM exchange in estuary ecosystems is largely unknown. But the salt marsh is very important. So because salt marshes are located at the interface between terrestrial and oceanic ecosystem, and salt marsh have high primary productivity, and surprisingly in our salt marsh site, the net primary productivity is similar with the forest ecosystem. And also salt marshes have high biodiversity, provide critical ecosystem services, and also with high socio-economic benefits. So yeah, it's very important to see how the GM exchange in the salt marsh ecosystems. Even there are a few studies before, but none of them is spending the full year. And in our study, our goal is to determine atmosphere surface exchange of GM in a densely vegetated salt marsh in estuary in Massachusetts, USA. And here is a map showing the study site, Plum Island Sound Estuary Salt Marsh in Massachusetts, and the red dots indicate the tower site. And also, we want to investigate the magnitude, seasonality, and the diel pattern of night GM exchange, including the deposition and the emission. And here is the method we're using to measure the GM exchange. This is based on tower-based micrometeorological flux gradient measurement, and we are going to quantify night atmosphere surface exchange of trace gases, such as in our case is GM, gaseous elemental mercury. And you can see that we have 2 samplers. One is at the 15 meters, and the lower one is at around 11.7 meters. And also we use the sonic anemometers to measure the turbulence. And we are going to measure the night ecosystem GM exchange, and it's including all the deposition, such as plant uptake, and all the emissions, including evasion over water surfaces over the whole ecosystems. So you can see in this picture, we have a solar panel because we don't have a power there. So that is really challenging for us to set up this instrument. And also the really bad things happened in the first year of our analysis because the tower was collapsed due to the heavy winter storm. And we are installed the tower again, the mid of July of 2022 because of the supply chain of the pandemic, everything is messed up. So we have to delay our measurements. And this is the equation we're using to calculate the GM fluxes. The method is based on the Edwards in 2005. So you can see that we have the 2 gradients. That is a delta C, and the other parameters are analyzed from the sonic anemometers. I don't want to include too much information about the equation, but this is the thing we're using to do our calculation. And next thing, I'm going to introduce my results. So the first graph, figure A, showing the 30 minutes GM fluxes, and the x-axis showing the sampling month, and the y-axis showing the mercury fluxes. And you can see that we have different color indicated different years. So the blue color indicated the first year analysis, that is 2021. And the red color indicated the second year analysis of 2022, but is from the mid of July to the end of the year. And the third year, I use the green color indicated, that is from the beginning of this year to the September. And you can see that everything above the zero line I indicate emission, and everything below this line indicates the deposition. And also it's really obviously to see we have really high variability, and it's a range from negative 500 to the positive 500 nanogram per square meter per hour, with the average of net emission, small net emission of 0.5 nanogram per square meter per hour. So the high variability also indicates it's really challenging to measure the GM fluxes in the salt marsh ecosystem with this method. And in order to better understand those data, and we add up all of these dots by years together. And so that is the figure B showing the cumulative GM fluxes. The same thing, the color indicate the different years, and the x-axis showing the sampling months. So it's kind of really hard to find some repeatable and overlap patterns, and almost no clear seasonal patterns as well. But one thing we can notice that at the mid of July to the mid of August is kind of they're showing the mercury emission. And in order to better understand those data, what that means, so we further separated those graphs by different periods. So the figure A shows the first year of analysis, which is from May of 2021 all the way to the January 16th of 2022. That is before our tower collapsed. So we separated into 4 periods based on the general patterns. And for the second and third year analysis, because that is a continuous analysis, even there are some gap there because of the data is kind of questioned. So we just delete some of the data, but we put the second and the third year together. That is from the mid-July of 2022 to September of 2023. And also based on the general pattern, we separate into 7 periods. So we have 11 periods total. And then we analyze those data by periods. And the next graph showing the diol patterns of GM fluxes of selected 4 periods, because the general pattern diol patterns are pretty similar of these 11 periods. So I just randomly selected 2 in the first year analysis and 2 in the second year of the analysis. So you can see here the x-axis is showing the hour of the day, and the y-axis is showing the GM exchange fluxes. So the GM fluxes even shows very high variability, but they still have diol patterns. We can see there are smaller and less variability during the nighttime, but high variability during the daytime. The high variability during the daytime also give us the indication that the flux during the daytime drive the general cumulative GM flux patterns. And what is the factors drive those patterns? And we haven't get a really clear conclusion right now, but at least based on the previous studies of forest study, we try to combine with the carbon dioxide and vegetation activities. And before introducing our results, and I want to introduce results of mercury with these factors, I want to introduce carbon dioxide fluxes and normalize the difference vegetation index, which is NDVI first. And in this graph, the same thing, the figure A showing the first year of analysis, and the figure B showing the second and third year analysis. X-axis showing the months and year, and the y-axis showing the cumulative carbon dioxide fluxes. And the red color indicates the carbon dioxide fluxes, and the green color indicated the NDVI. So in here, we can see that during the growing season, from May to October in this salt marsh ecosystem, the cumulative carbon dioxide fluxes normally showing the decreasing pattern. It makes sense because that is kind of plants are really high activity, so it uptake carbon dioxide from the atmosphere. And also during this time with high NDVI, because the NDVI is normally used to assess the health and the density of the vegetation in a specific area. And we can see the similar thing in the second year, the decreasing cumulative carbon dioxide fluxes and high NDVI value, and also the third year. And then we try to apply those two factors with the cumulative mercury fluxes. And let's see the cumulative GM flux with carbon dioxide fluxes. So the blue color indicated the cumulative GM fluxes, and the red color indicated the carbon dioxide fluxes. So in here, we can see that during the active growing season, the GM fluxes, which is the blue color of the first year, you can see the figure A first. So you can see that some period of time shows kind of corresponds well with the carbon dioxide fluxes showing decreasing, and some months shows emission, and sometimes showing decreasing and the emission again. I think it will be easier to see with the laser point. So you can see decrease, increase, decrease, increase is not the coupling well. And we can see that the second year and the third year, and during the growing season, some months showing the opposite pattern, and sometimes it's showing the similar pattern. But the third year is more like the totally opposite patterns. So from this figure, we can see that in our self-marsh ecosystem, there is no significant growing season vegetation GM uptake because of the decoupling patterns of cumulative GM fluxes and the carbon dioxide fluxes. And then let's see, then let's see how the cumulative GM fluxes and the NDVI. So I still use the similar color to indicate a similar thing. So when we see the growing season with a high NDVI value, so some months showing decrease and some months showing increase, decrease, increase of the cumulative GM fluxes. And but when we see those 3 years together, but one thing we can notice, and also we showed in the first result slides. Okay, I think, yeah. During the midsummer time, the GM shows emission. That is the thing, because during the greenness and with high NDVI value, that is we found. But yeah, that is not the plant uptake. So our conclusion is still kind of solid, that there's no significant growing season vegetation GM uptake. And in order to better to understand or support our results, I also use the vegetation results from our previous study to support our observation from the GM fluxes. And before introducing our results, I want to include some background information about the stable mercury isotope signatures. So these methods are really powerful to trace the mercury sources. And it includes 7 isotopes of the mercury. For both mass-dependent fractionation, I use small delta 202 mercury to show here, and the mass-independent fractionation, that is, I use the capital delta 200 mercury. So these two isotopes are kind of abundant. So that's the reason I use these two isotopes to show example here. Briefly information about mass-dependent fractionation, that is caused by the kinetic process, and one of the largest MDF process is kind of foliage uptake lighter mercury from the atmosphere than the heavier mercury residue in the atmosphere. So which indicates, which caused the kind of a larger delta small 202 in the atmosphere, but kind of more negative and smaller small delta 202 in the foliage. And for the mass-independent fractionation, MIF, which is most caused by the nuclear volume effect and the magnetic isotope effect, and the photochemical reaction, that is the process can impact the MIF values. Well, overall, I want to highlight the two important things in this slide. First of all is mercury signatures different in atmospheric species. You can see that atmospheric GM and different signatures with precipitation oxidized mercury. And also the recent research have found that around 90 to 100% of forest leaf mercury originates from atmospheric GM uptake. So that is in here, uptake from the atmosphere. So how our salt marsh vegetation looks. Then I overlap my results salt marsh vegetation signatures in this figure, and this part of the information have been published, but it's still under last step of the review, but I want to include in here. And we can see that the salt marsh foliage signatures I group with the green color here, and it shows distinctly different to that of forest foliage with less negative small delta 202 and more positive capital 200. And our mixing model results indicate our salt marsh vegetation is kind of mixed sources of atmospheric GM around 32%, precipitation oxidized mercury that is around 33%, and the rest of 35% from the vegetation roots uptake, which also to interpret it that salt marsh biomass mercury sources are not dominated by the atmospheric GM uptake. So the results from the salt marsh vegetation kind of consistent with the GM measurement, the lack of vegetation GM uptake. So here's all of my presentation, and I'm going to conclude my fund. So the GM flux patterns show high variability with almost no clear seasonal and repeatable patterns. And also GM fluxes show the dial patterns with a smaller and less variable during the night and high variability during the day. And we don't detect significant growing season vegetation GM uptake because of the decoupling of GM fluxes and the carbon dioxide fluxes. The apparent seasonal lack of vegetation GM uptake is consistent with our previously marsh vegetation isotope results in these ecosystems. Possibly very high surface variability, such as high and submerged marsh, tides, water surfaces, and channels make the detection of low GM fluxes using tower-based methods in these ecosystems very challenged. So we are still working on what the dominant factors impacted those patterns, but yeah, that is our next step. Yeah, that's all of my presentation today, and I'm happy to take any questions you have.
I have a question about that variation, like during the growing season, where you saw some go down a couple slides.
Okay.
Like when you were just saying that, like, I think so like in that, on the left side, you have like the up and down of the deposition or level of flux. Did you compare that to precipitation events? Like, was there like kind of drought that may have caused that, or flooding maybe in the wetland?
That is a really good question. Actually, when we analyze those data, we filter out those data with the precipitation effect, because I think that is the kind of cannot tell the general patterns of it. But for the tides, we analyze it a little bit for the data in that period, but we didn't find very interesting story so far. But we still, those data will be very detailed and try to find some story behind it.
Thanks.
Yep.
Great talk, Ting.
Thank you.
I just had a quick question regarding if you looked at the concentration trends at all. Do you see any sort of seasonality in concentrations, or is it also very noisy?
For right now, it's kind of very noisy, but one pattern I can tell that I think for the data during the summertime, we have large variation. For the seasonal patterns, I think it's kind of hard to see for others. Yeah, that is what I can see from our current data right now.
Thanks.
You're welcome. Thank you.
Our next speaker is Eric Roy from UMass Lowell and MIT.
Well, thank you very much. That was, it's going to be difficult to follow Ting. We've worked together a lot at UMass Lowell over the last few years. But today I'm going to be talking a little bit about work I did as an undergrad there, primarily looking at two forest sites in New England, Harvard Forest in central Massachusetts and Howland Forest in northern Maine. And at these sites, we measured gaseous elemental mercury dry deposition, or, well, the bidirectional exchange. And basically, the main point of this work was to say, how much information can we get out of just looking at concentration variability that we measure from Techeran measurements, so active measurements that are at relatively high temporal resolutions, as well as passive samplers. So kind of just to dive in, why would we even think that we could do this sort of analysis? And hopefully this curve looks a little familiar to you all. Obviously, this is the Keeling curve. And so we have this 60, 70-year record of carbon dioxide concentrations at a background site that is able to show signals, right? We're able to see hemispheric scale emissions in this trend. We see an exponential increase in carbon dioxide concentrations due to anthropogenic influences. And we also see this nice seasonal sawtoothing pattern associated with the seasonality of net ecosystem productivity. And there have been studies that have shown that you can get some sort of quantitative information out of this. So one might think that if Mercury is doing something similar to carbon dioxide, maybe we can do something similar. And people have looked at this. Yiskra et al. In 2018 were able to show, through a meta-analysis of mercury concentrations, were able to show that there was a positive, significant relationship between the amplitude of mercury concentrations at a given site from year to year and that of carbon dioxide. So basically, your carbon dioxide decreases by this amount, and your mercury concentration will scale with that. However, there was a follow-on study by St. Louis et al. The following year that looked at 10 years of data from the experimental lakes region or the experimental lakes area of Northern Ontario, and made the argument that maybe this is more complex than just some sort of linear trend. On the right, we have these three-dimensional plots. Can you see mine? Yeah, you can. Okay. So on these 3D plots, you have your seasonality on this axis, you have your diurnality on this axis, and if you're trying to make some sort of case that the ecosystem is controlling your concentration of mercury, then the difference between these two plots, that being your gross ecosystem productivity and your concentration, would suggest that maybe this isn't the case, right? So net ecosystem productivity is maximized during the summer, during the day, and then at night, it's pretty much flat. I should say this is gross ecosystem productivity. So they suggest three reasons for why these patterns diverge, these being either the nocturnal deposition and vertical mixing associated with mercury, maybe a revolatilization of mercury from surfaces in the forest, or potentially a bidirectional stomatal exchange. And so we want to use these two studies that we worked on in New England to try and figure out, well, first, do our fluxes imply this uptake that's been presented by numerous studies at this point? And if so, how do we reconcile this with the diverging concentration patterns with that of CO2? Then we also seek to figure out if we can use these concentrations to figure out what's going on with our ecosystem exchange. And then can we use passive samplers to get at the same thing? Because that would be a lot cheaper. And so the study sites that we'll be talking about, again, Harvard Forest located here in central Massachusetts in a relatively rural area, and similarly with Howland Forest in northern Maine. And they've been measuring ecosystem scale fluxes for years and years and years at these sites, two of the oldest sites in the world. So it was a real honor to be able to do some measurements at these locations. And so similar to Ting's discussion earlier, so I don't have to go too in depth with this, but we use this aerodynamic flux gradient method that basically says you have some sort of gradient in your concentration, you have some sort of eddy diffusivity, you take their product, and then you can understand what's going on with your flux. And we were able to tune that eddy diffusivity to match concentrations or fluxes of carbon dioxide that were measured on these same gradients, and compare those to eddy covariance measurements, so we could kind of tune to make sure that our parameterizations work out. And so we measure above the forest, we measure above the canopy, and we're measuring our concentrations of mercury using some TechRain analyzers with a 5-minute concentration resolution, and by the time we do our switching between inlet lines, we get a 30-minute flux resolution. So at Harvard Forest, we measured from 2019 through 2020. And then at Howland Forest, we did the same thing pretty much for the TechRain measurements a year later, beginning during kind of the peak of, or just after the peak of COVID. And then we also included these little blue things on the right-hand side. And these are our passive air samplers, MERPAS samplers that were provided by Frank Venia. And we deployed these for 1.5 to 3 months at a time and did a total of 98 samples from January 2021 through May of 2022. And so the first step was to kind of figure out, okay, how do carbon dioxide patterns compare with our mercury patterns? And so as I had mentioned before, the way we're going to read these plots are from, on this axis, you're talking about your seasonality, and on this axis, you're talking about your diurnality. And basically what we can see is that in the winter, your concentration is pretty flatlined, not a lot going on in your ecosystem, pretty much expected. During the summer, you have your minimum concentrations during peak day of carbon dioxide at both sites. However, you also have your maximum concentrations occurring at night. Now this coincides with a, this coincides with a flux pattern where, which is what we would expect, right? And I should also note that the blue colors represent deposition, yellowish colors represent emission. So during your daytime, during the active season, you have a lot of drawdown of carbon dioxide at both sites, and then at night you have a slight emission. So, you know, at night you have respiration, you don't have deposition. What's interesting is that your magnitude of your flux at night is relatively small compared to your daytime deposition, but you have these really high concentrations of carbon dioxide. So there's something else at play besides just your flux. Your stability also becomes important. We'll talk about that a little bit more in the case of mercury. So for mercury, again, looking at the seasonality, generally speaking, higher concentrations in the winter, lower concentrations in the summer, similar to what had been argued in previous studies. Your amplitude of your diurnality is maximized during the summer and minimized during the winter, again, similar to carbon dioxide, except going back to what was originally alluded to by St. Louis et al., the direction of this diurnality is different, right? You have your maximum concentrations during the day and your minimum concentrations at night. So what do we actually get for exchanges? Well, we do get a deposition that's dominated by daytime deposition during the active season at both sites. Now the timing of this is a little bit different, but what you'll notice is your maximum deposition occurs during midday during the summer at both sites. You have a tendency for emission during the winter at both sites, and during the active season you also have a slight tendency for deposition throughout the day-night period. So unlike carbon dioxide where you have this switch in which direction your flux is going, your flux is always negative for mercury, which is consistent with what other studies have shown. So we could say that yes, the seasonality of mercury fluxes do coincide with what we would expect for carbon dioxide or other biogenic or biologically active trace gases like carbon dioxide. And this is especially true during the day. And so again, it supports this dominant vegetative control on mercury. Now, let's forget about the diurnality for now and then just kind of look at things on a daily scale. The blue curve are the cumulative flux for both sites. The green curve represents a 60-day centered rolling average of your daily average mercury concentrations at both sites. And then the other dots represent either a 7-day average with this dark one or the daily average in the teal colors. And basically what you can see is that during the late spring, you have your maximized concentrations, you have a tendency for a trend in emissions, and then this switches as your ecosystem starts to begin assimilating carbon dioxide. If we then look at mercury, we see something fairly similar. we see that the mercury tends for an emission throughout the winter, and then right at the beginning of your active season, you start seeing this trend downward. And this coincides very well with the timing of your seasonal drawdown of concentrations of mercury. And this is consistent with both sites. So I guess the main points to take away from this are that the onset of cumulative deposition is very well aligned with the onset of your seasonal concentration declines. So that might be a pretty good proxy of figuring out what your ecosystem is doing if this is the case when you have these dominant signals. We see that there's some subseasonal variability captured at different sites, right? So we're not seeing a hemispheric signal because at Harvard Forest, we see this interesting dip that occurs in late winter. And then this coincides with a dip in the cumulative flux as well. So we're seeing things that are local scale in these concentration trends. Now, it's also worth noting that the flux magnitudes, so the amplitude of your blue line for carbon dioxide and mercury scale between sites, right? You have higher at Harvard Forest, lower at Howland Forest. However, your concentration declines don't. So this indicates that there might be some more complex factors at play if you were trying to do some sort of quantitative analysis of, you know, your concentration goes down this much, your flux must be this much. So we get really good agreement in the timing of our fluxes and our concentrations. However, we also see, and I didn't allude to this well enough, the timing of these late season reversals is not as well aligned. And this will again be impacted by the stability of the system. And so now bringing in these passive measurements, on the top here in these box points, these are our passive sampler concentrations. On the bottom, these are our active TechRAN measurements. And basically, we average our TechRAN concentrations for each of these levels over the same period at which we measured using the passive samplers. And what we can see is that the seasonal variability of both measurement systems does really well. The timing, you have your winter concentrations are high, your summer concentrations are minimized. The magnitude of your seasonal declines is pretty well captured, you know, about 0.4 nanograms per cubic meter. So that's really promising. We do see that the vertical distribution of where these points line up, so the soil points, these being the brownish colors, and the above canopy points, these being the teal colors, we see that these are largely consistent, but because of uncertainty in the measurements of the passive and active measurements, we weren't able to do kind of a statistical test on this. Now, the offset in magnitude that you may notice between these is likely due to some instrumental biases. With these studies, our main goal was to make sure we didn't have any bias in our concentration gradients. We wanted to make sure that our concentration gradients were not exhibiting some sort of bias that would influence our flux calculations. So while we did do permeation injections at the beginning of each study and at the end of each study, there is the potential for some sort of bias in our measurements. And so with that, the question becomes, okay, so the passive samplers do a pretty good job of figuring out what the seasonality looks like. Can we get anything out of the underlying exchange from measuring these gradients at the same height? And we kind of propose a theoretical limitation to the use of passive gradients for flux inference. And the reason for this is when you use a temporally integrated gradient, you're going to be biased towards your nighttime periods. So here we're looking at Harvard Forest. On the left, we have our cumulative carbon dioxide flux, and that's partitioned into day and night. So the gray is your overall, blue is night, and yellow, which I hope you could see. Yeah, it came out good. That is your day. So during the day, you have a lot of deposition, as one would expect. At night, you have this tendency for emission due to respiration, and overall, it leads to deposition. But if instead of looking at this entire flux equation, you instead say, well, let me just look at this concentration gradient and look at it over time, you see that your overall implies a large emission, and you're dominated by what's going on at night, which isn't terribly surprising. You need turbulence to figure out fluxes. If you look at Mercury, we see something similar, but because you have this difference in the direction of the flux at night, you have a slightly different pattern, where instead your cumulative concentration difference is, again, dominated by nighttime periods, but it does still indicate that you have some sort of or implies deposition, right? Because the only thing that can change the sign in this equation is the concentration gradient. But this kind of brings up the issue that while you might be able to get something that scales, you might be getting the right answer for the wrong reasons because of this theoretical limitation. And so kind of just to drive this point home, at night, you have carbon dioxide emissions, and you have a slight mercury deposition. This implies a vertical profile of concentrations in which your above canopy concentration of carbon dioxide is slightly higher than the background, and your mercury concentration is slightly lower than the background. When you start, when the sun comes up, you start mixing the atmosphere and you switch where these fluxes are occurring. And so by mixing the air aloft and also switching the fluxes, you end up getting a carbon dioxide concentration that declines at the surface and a mercury concentration that increases at the surface relative to its nighttime pattern. And so overall, because even though you have these dominant deposition forcings, you would get, or you at least would expect, a diurnal difference in your concentration time series. And so with that, I leave you with a few summarizing points. This work was recently published in Chemosphere, so if you're interested, please check out the QR code here, and I'm happy to field any questions. Thank you.
Hi, Eric.
Hi.
Thanks for participating in the MELD Science Committee yesterday. And you mentioned you used the passive samplers at 1.5 to 3 months duration, and I was wondering what your experience with that was, and if you have recommendations for NADP moving forward with a national network.
That is an excellent question that I wish I had a more rigorous answer to. In our case, we were able to, for that integration period, I don't think we had any large, we weren't below the detection limit on any of our samples, which was great. I wasn't actually involved with the actual deployment of these samplers. That was taken on by Jun Zhu, as well as Daniel Oberst and Frank Vania. So I would need to maybe consult with them a little bit more. I'm sure Frank, and I'm sure you might be in contact with him as well, but I'm sure he'd be able to provide a more rigorous answer on how much time you actually need to get something that goes above your detection limit for these samplers.
Sounds good. We'll follow up with you.
I have a question here. Great talk, thank you. Quick question on the comparison between the TechRANs and the MERPAS.
Yes.
So you mentioned, I was just wondering if you could elaborate a little bit on the the inability to account for the uncertainty to do like even a regression to test the two, because it seems like you could actually get to that theoretical maximum by looking at the slopes of that relationship potentially. And I just didn't know if you had any insight on that.
Yeah, that's a good point. I think one of the issues with doing this sort of analysis that you kind of keep running up against, and I think we could see it with some of the other plots, or that the measurements are just really noisy. And so I guess trying to use a regression technique for this would be ideal. The question just became, how do we rigorously define what our uncertainty is for the active measurements and the passive samplers. And the main reason why we didn't necessarily go down that road even more was the offset that we had in our concentration. So originally we were trying to figure out, okay, well, can we start parsing out, oh, during this period, everything lined up well. During this period, everything lined up well in the vertical distribution. And when we started going over what the uncertainties of the passive samplers were in our own measurements, it didn't really make sense to keep pursuing that. But I think more work in that area could be done. Yeah.
There's one question over there.
Okay. Very nice talk, Eric. I have a question. Do you have some physical explanation for the point 1 of your conclusion? What's the underlying physical reason?
The physical reason?
Yes.
Yes, for one, I would say it's dominantly vegetative uptake. And this has been confirmed by previous studies that have, as Ting had alluded to, that have done kind of the isotope measurements to confirm that these fluxes are indicative of vegetative uptake. However, that being said, in June's recent paper earlier this year, he was able to kind of show that differences between sites are dominantly impacted by differences in what's happening at your canopy level versus what's happening at the soil level. So, for instance, he was able to explain that to a certain extent, the difference in your cumulative flux of mercury between Harvard Forest and Howland Forest was largely due to an emission from the soil at Howland Forest, which contributed to a lower overall ecosystem flux. So the fluxes that I was showing here, we're just looking at that above canopy gradient, and so we're just looking at the total ecosystem and not saying, okay, well, what's the soil doing and what's the canopy doing?
Okay, thank you.
Yep.
Thank you, Eric.
Okay.
Our next speaker is Isabella Garriac. I hope I pronounced her last name correct, from Umel University.
Hi,
Take it away.
everyone. My name is Isabella Garriac. I'm brand new to NADP, so I'm really excited to be here and be able to talk to you all. I'm a current Fulbright Research Fellow at Umeå University in northern Sweden, so it's currently pretty late for me here in Sweden. But this project is not really associated at all with the work that I'm doing here. I did my bachelor's at Macalester College, and my senior capstone project, I partnered with the University of Minnesota and the Spruce Project to work on this. So this is some of the results that I have had so far from my bachelor's capstone. Title being Understanding Mercury Gaseous Fluxes at the Marcel Experimental Forest and the Spruce Experiment in Grand Rapids, Minnesota. So I realize that I'm talking in a session all with a lot of people who study mercury all the time. So I'm sure a lot of this will be a bit of a repeat, but I think it's really important to talk about the background of why we decided to look into this. Mercury is a contaminant that's found in every ecosystem. And there are many health effects that we've seen from mercury, which some of them are listed to the left here. And I definitely in my lifetime have seen mercury go up in the media, and it's becoming very well known to a lot of people that it's something that's pretty dangerous and is considered a major global concern. And most of this atmospheric mercury that we're seeing is or deposition of mercury is related to human emissions, around 70%. The other 30% coming from natural events like volcanism and wildfires, but we are definitely part of the system there. And pools of mercury in the atmosphere have gone up by about 300% since pre-industrial times, and pools in organic soils by about 200% since the same time. And in the atmosphere, it's present as gaseous elemental mercury, particle-bound mercury, and gaseous oxidized mercury. When I first started my project with Spruce, I thought that some of these health concerns were related mostly to inhalation of mercury, but it turns out that I learned a lot on this project, and we're a lot more worried about redeposition of mercury into waterways, which makes it more accessible to different organisms and is able to bioaccumulate through the food chain, which then leads to plant, animal, and human health consequences. And Kevin Behrens, who is the master's student who is also working on this project, he focuses more on plant uptake of mercury. So that's an interesting addition to this as well. And then climate change is another global concern that I'm sure we all know about and are very scared of. And the IPCC reports that at the current rate of human activity, we're likely to see an increase of over 3 degrees Celsius by the year 2100. So all of these things coming together make this something worth looking into, mercury and climate change. So I am really interested in looking at peatland ecosystems. The image to the right is a picture that I took actually up at Spruce that has a very classic peatland look to it with the dark organic matter in the water flowing off from a peatland system. And in peatlands, mercury is released into the atmosphere through chemical and physical transformations by microorganisms. And usually when I talk to people who are not in the sciences about how they do this. It's like, well, the happier the microorganisms are, the more that they're living and eating things up and then sending gases out into the atmosphere. And then they're able to convert mercury from its inorganic into its organic form. And peatlands are also particularly at risk from climate change that I mentioned earlier. And that's a worry to people because of many of the ecosystem services that they provide. Beyond being able to trap mercury, they also conserve biodiversity, minimize flood risks, and also store CO2. So that is, yeah, I was particularly interested just in these systems, but they seem to have a big relationship with mercury as well and climate change. So this is an image on the left of me and my main supervisor, Dr. Randy Kolka from the U.S. Forest Service, when I was able at the end of my senior year of college to go up to Spruce and really see the whole experiment, which was really exciting. This is us in front of one of the enclosures that I'll talk about in a second. But the main objective of this study was to determine whether increased temperature in peatlands will contribute to atmospheric mercury, and also secondarily, if increased carbon dioxide will have any additional impacts on those fluxes. And my big research question is, will different temperature treatments result in peatlands acting as net emitters of mercury? And I had kind of an interesting introduction into working here because I was just sitting as, I don't know, a sophomore in college in my climate systems class, heard about this really incredible project that I was shocked I'd never heard of before, where they were studying climate change on a pretty large scale that was basically in my backyard in Minnesota. So I reached out just thinking, I really want to be a part of this in some way. And then the Mercury team took me on and we were able to collaborate together. So these are some images that I also took on my trip of some of the enclosures that there are at Spruce. So Spruce itself is an acronym. It stands for Spruce and Peatlands Responses Under Changing Environments Project, and it's located at the Marcel Experimental Forest, which is just northeast of Grand Rapids, Minnesota. That's about 6 or 7 hours north of where I was going to school in the Twin Cities. And within this experimental forest, there are 10 warming enclosures, which you can see an image of one of those 3 images all together are of one of the enclosures at this site, some from the outside, some from the inside. It was really cool to be able to see them. It was walking around, it was like being on another planet. And I thought it looked really amazing to see them all there and all the work that was going into maintaining them too. And so in those 10 enclosures, there are 5 different temperature treatments that go all the way from ambient 0 degrees Celsius of warming all the way to 9 degrees Celsius of warming. And like I said, there are 10 enclosures, but 5 treatments of temperature. All of them have a pair. So there would be 2 ambient plots, 2 that are at 2.25 degrees Celsius, et cetera. And 1 is just treated with additional temperature, and 1 is also has elevated carbon dioxide. And when elevated with carbon dioxide, they usually try to keep it around 800 parts per million of carbon. And there are some additional sites as well that I thought I would mention, including an NADP site. So that's interesting as well. The Project Spruce, like I've already been singing its praises, it's really cool. And there are a lot of people looking into a lot of different things related to vulnerability of organisms, biogeochemical processes, and ecosystems to climate change, and also limits to regeneration, and even more. So for the experiment that I was looking into, we, like I have already said, we're looking at temperature's effect on mercury fluxes in northern peatlands. Even though it's already been mentioned a few times, I threw my definition, or the definition of mercury fluxes up there, of mass of mercury emitted by 1 square meter of soil per hour. And we also use passive air samplers. I'm really glad that I went right after Eric, because we also were, I was exposed to these for the first time here. And we use deployments using passive air samplers as well. So we measured atmospheric mercury concentrations, examined based on the conditions at Marcel using these samplers. which were collected every 3 months. So we had 4 deployments per year, so kind of like a seasonal collection. And I was not part of the actual sampling that happened. I came in when they had already finished the 2021 data. I think they were still collecting some of the 2022 data when I came into the project. And so there were some calculations that we did at this point, because you can't just measure mercury flux very easily. This is a measurement of concentration. So first we calculated the mean mercury concentration in the enclosures. My data was all from the years 2020 and 2021. And then that was converted into atmospheric concentration of mercury in each enclosure over the deployment using an equation from McLagan et al., which also included wind speed as a factor, because as you can see in the previous image, the enclosures are all open to the air, which means that they're able to have rainfall, snow, organisms, and wind all impact things that are going on the inside. Oh, I also forgot to say on this slide, but the image to the left behind the text box is one of our ambient enclosures, which doesn't have any walls around it at all. So all of those are just open to the air. Yeah, so my data, I had 1 point per enclosure per deployment over this 2-year period. It was a little bit of a limited data set, especially since we would have had more data from the end of 2019, but unfortunately, some of it got lost. So I would like to have more data to work with, and they're still collecting it now. So hopefully there'll be more to look at in the future as well. So these are some of my preliminary results, and I'm still working on making some graphs for some others. I looked at mercury flux compared to air temperature, as well as soil temperature at 2 different depths, 10 centimeters and also 20 centimeters. And I found significant linear relationships between mercury flux and all 3 of these temperatures, although it looks like air temperature explains more variability than the soil temperatures do. And you can see my R² values there. And we saw highest mercury fluxes at over 20 nanograms of mercury per meter squared per hour. So there was a pretty big difference by the time that you were getting up into those higher temperatures. So just to summarize that finding, using passive gaseous mercury sampling, we found that temperature was a covariant, which showed a positive correlation to flux rate over time. Air temperature explains more variation in mercury flux than soil temperature. So this was a pretty significant finding, especially since we are fairly certain that there will be temperature changes happening in the future due to climate change. And especially in peatlands, which occur mainly in higher latitude environments where climate change affects ecosystems at an especially fast rate. So even if there's a global temperature change of around 3 degrees Celsius, we don't really know what that change is going to look like in some of these northern latitudes. So that's something of particular importance, which is why you see those incredibly high temperature changes on the Spruce project. I think most of the people working there really want to see, they reflect predicted global warming values from the IPCC, but also much beyond those predicted values, because they really want to see how those ecosystems are being pushed to their limit and even beyond their limit. Something that I immediately think of is, at a certain point, when are these deposits of mercury going to be used up? We don't really know how to say that with the information that we have. But definitely when you get into those much higher temperatures, I'm not really sure how big the deposits would be by that point as well. But understanding mercury cycles is critical to our understanding of how things are going to change in the future. And also because it's such a medical concern to humans and a health concern to plants and animals and our ecosystems. I think this is something definitely worth looking into more, and also adding that to our general knowledge base about peatlands and how they're affected by climate change. The images at the top as well here are just showing you the even larger scale, some drone footage of the Marcel forest, where you can see some of our ambient plots as well and just what those look like in the general scale. And I'll close my discussion by saying that I have a lot of gratitude for this team and being able to participate in something, especially at such a young age. I only just finished my bachelor's now, and to already have found something to me that's this significant was really incredible. And for future work, there are a couple things that I'm still looking into myself, like including carbon dioxide in my current data sets analysis. And also, while I was looking at my data just from a visual, I haven't really done the statistics on it yet, but seasonality seemed to be something that could be interesting to look into as well, which was also just in the presentation right before me. So maybe I should chat with Eric a little bit more about it. And then beyond my study with a larger data set, I think it would also be interesting to look at mercury gaseous fluxes in other ecosystems and also across longer timeframes, because having 2 years is great, but I think also being able to see how those temperature changes would happen even longer over time would also be really valuable. And I'll just wrap up by thanking Dr. Randy Kolka, Dr. Ed Nada, and Kevin Behrens for their help on this project. I'm really thankful to be a part of the team. Dr. Carl Mitchell for helping to develop our samplers and also for processing a lot of our samples at his lab at University of Toronto. And Gopinski and Cole Stenberg for their help with sampling, Dr. Paul Hansen for leading the Spruce project, and also Dr. Christy Manning, my academic advisor at Macalester College, in helping me in my last months of my undergraduate degree. And with that, I say thank you and happy to take some questions.
Questions for Isabella?
I'm not an expert on Mercury, so hopefully this isn't a stupid question. But I'm thinking about the two talks we just heard. And the first talk, we heard that there was general correlation between CO2 levels and the diurnal pattern with Mercury, and that there was some kind of, I didn't really hear too much about, well, what is the physical process that would explain why there's different levels of mercury absorption at night and during the day? I mean, to me, as a plant physiologist from way back, the only difference I can think of in the plant would be the stomata are open in the day and not at night, so there could be more influx during the daytime. But then we just heard, well, temperature is very important. So could this just be a physical phenomenon in terms of the day and nighttime differences as well? It's cooler at night than it is during the day. I mean, I'm just trying to understand what's the physical basis for why there would be different levels of uptake by the vegetation at night than during the day. So
that is a really interesting question. I'm not sure that I have the background to be able to answer it. I'm just thinking as well that I'm also curious about that. Like I said, I think seasonality appeared to have an effect, and that would be for the same reason that temperature would be different during the colder seasons, and that was where we were seeing less of a slope or a flatter slope for mercury flux change. I'm not sure. I'd have to look into that more.
Eric's still here. I wonder if he has any thought about that.
Thanks. Yeah, it's a really good question. Great talk, by the way. I think the main point that I was trying to make was that what you're measuring for concentrations depends a lot on what your turbulence looks like. As far as why your fluxes look like the way they do, there have been studies that have shown that dew uptake of mercury in the morning is one potential pathway for deposition during the night. there's a number of other pathways that are kind of floated around. I don't think it's really well constrained why you have this nocturnal deposition of mercury. But then the question of how temperature links in with this is a really interesting question that I guess I haven't looked into much either. But yeah, not a great answer, but thanks.
Can you hear me? I think I can add up a little bit. Yeah, because I think if you just look at the
Hello?
vegetation, for example, the forest or
T-
yeah, foliage
could-
itself, I think
Hello?
yes, right. The warmer temperature will make the plants because grow more actively. So the stomata open, yeah, and can uptake more mercury from the atmosphere. But if you just see the whole ecosystems, for example, the salt marsh ecosystem I'm looking for. So not only the vegetation is the important reason, and also other the microbes in the ecosystems, especially the peatland and also the salt marsh. There are so many microbes there near in that ecosystems. So that is another reason that will be impacted. So yeah, that is my answer for that question. But I'm still learning on it. But that is my understanding. And also in my case, I said, oh, there are some factors will impact the general mercury patterns, and the temperature is important also. But I didn't show it here because it's kind of complex as well. I didn't show the high temperatures, the warmer temperature shows continuously mercury emission or the larger, yeah, just a larger variation in my case. So I think different systems, I think it will show the differently. But yeah, general, the answer is that warmer temperature will make a vegetation uptake more mercury from the atmosphere because of the stomata activity. Yeah, that's my answer.
Thank you for that. And thanks, Isabella, for your talk. Great work. Okay, next up is Guirong Xu from National Central University in Taiwan. Good morning, Guirong.
Good morning. Thanks, Dave. Yeah, so this is Guirong Xu giving the presentation from Taiwan. So good morning. It's 13 hours ahead of Madison time here. So I would turn your attention from North America to East Asia temporarily during my presentation to explore the influence of climate on atmospheric mercury concentration at a mountainside in Taiwan. So previous study have already indicated that atmospheric mercury cycling is sensitive to climate-derived change. However, the links with various teleconnections are not very well studied. It's mainly because due to the lack of long-term monitoring data set of atmospheric mercury. And in Taiwan, the Luling Atmospheric Background Station has started monitoring mercury in the air since April 2006, and it is still ongoing. So to date, we have already accumulated 17 years of atmospheric mercury data. So we analyzed this long-term mercury data set of Mountain Luling to explore the relationship between climate and atmospheric mercury variations. So Taiwan is located to the southeast of East Asia continent. And the Luling Atmospheric Background Station is located in central Taiwan with an elevation of 2,862 meters above sea level. The background station started operation since April 13, 2006, and the atmospheric mercury monitoring also started on the same date. So Taiwan is located in a region with significant monsoon activity. In winter, the Siberian high dominates, so there are more air masses coming from the East Asia continent to Taiwan. On the other hand, in summertime, the Pacific high dominate this region, so there are more air masses from southeast or from the Pacific Ocean to Taiwan. Therefore, because of the meteorological conditions during winter monsoon season, favor the transboundary transport of air pollutants, including mercury from East Asia continent to Taiwan. So we can expect some kind of seasonal variability in atmospheric mercury concentration here. And also, at high elevation, the westerly prevails. So from this map, we can see, so at the high mountain site in Taiwan, we could be seeing air masses from Indochina Peninsula or even from India to our mountain site. So the atmospheric mercury monitoring at Lulin Station, we use the Chakran speciation system, and we also collect rainwater for mercury analysis at the same site since 2009. But during my presentation, I will just focus on the atmospheric mercury. And the speciation system monitor concentration of gaseous elemental mercury, gaseous oxidized mercury, and particulate mercury. But for this presentation, I will just focus on the gaseous elemental mercury concentration variation. So in 2019, we published a paper to explore the temporal trend of mercury observed at Mount Lulin between 2006 and 2016. This is the time period before the Minamata Convention on Mercury entered into force in August 2017. And our analysis demonstrated that atmospheric mercury concentration was decreasing during that period of time, and the decreasing rate was 0.022 nanogram per cubic meter of air per year, or 1.5% per year. And this decreasing rate is in agreement with other published data from, for example, Capitol, Okinawa, Japan, or from South Africa, from Europe, and from North America. So this is interesting because the global emission inventory reported from UNEP saying that the mercury emission was increasing from 2010 to 2015. But we especially, and the increases in mercury emission is very significant in Asia. However, from our side, we are seeing the concentration is decreasing, not increasing. Okay. Also, we noticed that the decreasing trend somewhat disappeared between 2014 and 2016. And you may remember 2014 to 2016 is a very extreme El Nino event. So this stimulated our thought that this El Nino event or the climate variation may influence the concentration of mercury at our Mount Lulin site. So we explore the source-receptor relationship by using the backward trajectory analysis. So from the figure on the left-hand side, you can see that like usually during the springtime, we are seeing, and due to the influence of the westerly, we are seeing more air masses from the Indochina Peninsula in springtime. However, in 2015 and 2016, the air masses, the frequency of air masses from Indochina Peninsula dropped significantly, and it was replaced by air masses from the coastal area of China. So this again seems to indicate the influence of the El Nino event on atmospheric mercury concentration at Lulin. So we further analyzed our long-term mercury data set by using the ensemble empirical decomposition methodology to decompose the intrinsic nature of the raw signal raw data adaptively into single modes, known as the intrinsic mode functions, IMF. So we analyzed the data between 2007 and 2019, and a total of 14 IMFs were obtained by using the EMD with the periods of the signal ranging from 0.5 days to approximately 13 years. And IMF 3, 8, and S with a period of 1 day, 33.5 days, 182 days, and 364.1 days represent the diurnal, monthly, semi-annual, and annual GMN cycles, respectively. So in our previous study, we already know that the daily GM concentration variability at Mountain Lulin is mainly controlled by the local upslope movement of boundary layer air. That is the valley wind system here. And the seasonal variability of atmospheric mercury at Lulin is driven by the regional air mass differences in regional air masses, origin and transport passes. That is influenced by the monsoon system. So the amplitude of these IMFs represent the strength of each oscillation, and from the high frequencies signals, we noticed that the diurnal cycle was the most prominent among all the high frequency IMFs. And the low frequencies variability, that is the IMF 12, 13, and 14, was stronger than those of the local and the regional signals. So this highlights the importance of interannual variability of GM concentration and the associated driving factors. And IMF 15 was the residue. It represents the overall trend of the GM data set. So we can see the GM concentration decreased between 2007 and 2013, but it seems to increase between 2014 and 2017, then started to decrease again from 2018. So our group also analyzed the Southern Oscillation Index using the EMD, and we also got 5 IMFs for the SOI. And we also noticed that the IMF 3, 4, and 5 of the SOI have were similar in frequency or periods to the GEM interannual variability cycles. So therefore, we perform the correlation analysis between the IMF of GEM and IMF of the SOI. So we found they are correlated, but they are negatively correlated. So this correlation analysis revealed the dependency of the interannual variability of atmospheric mercury on large-scale climatology variations. Then recently, my master student further analyzed the data set, but this time, instead of using the whole year data, we focus on the data in March, because that's the biomass burning season in Indochina Peninsula. And that's also the season that the biomass burning emission could influence our Mountain Lulin sites. So the top panel shows the GM concentration of March, the mean GM concentration of March every year. And the bottom panel shows the mean NINO 3.4 indicators of March of every year. The NINO 3.4 index was used to indicate the El Nino or La Nina event. The value greater than 0.5 indicates El Nino, and the value lower than minus 0.5 indicate La Nina event. So from this figure, we noticed that usually the GM concentration are higher in El Nino years and lower in La Nina years. But we found the highest March GM concentration in 2017. This year is neither a El Nino nor a La Nina year. So because this analysis is focused on the influence of the like the large-scale air circulation on mercury concentration. So we first created the mean wind field at 700 hectopascal in this region. From the figure, you can see in March, the westerly prevails at the elevation about 3,000 meters above sea level. So this indicates during this in this month, we are seeing more air masses passing northern Indochina Peninsula and southern China before coming to Lulin Station. And sometimes the air mass could also be coming from India. Next, this figure shows the wind field anomaly between the El Nino years and the mean wind field. So we noticed that during the La Nina, El Nino years, the westerly enhanced. So I think there are more and more air masses from the source region, including India, Indochina Peninsula, and southern Taiwan before coming to Lulin. So I think that contributes to the higher mercury concentration we observed in El Nino years. And then this figure shows the wind field anomaly between La Nina years and the whole data period. And it's interesting to see from here that the westerly weakened and deviated from Taiwan. So in fact, we are seeing air masses from the east coming from the Pacific Ocean. So that's the relatively clean marine air. So I think this contributes to the lower mercury concentration we observed in La Nina years. And the year of 2017, the wind field anomaly shows that in addition to the westerly winds bringing the air masses from Indochina Peninsula and southern Taiwan, the air masses could also be coming from central and east China. I mean, that's another very important atmospheric mercury emission source region. So therefore, we think that's why we are seeing the highest atmospheric mercury concentration in 2017, although it's not a El Nino year or a La Nina year. So in summary, our study showed that the amplitude of the interannual variability of GM is greater than those of diurnal and the diurnal variabilities at Mountain Lulin. And we are seeing higher GM concentration in El Nino years and lower concentration in La Nina years. And in El Nino years, the westerly enhanced. So we are seeing more air masses coming from the source region, contributing to higher GM level. And in La Nina years, the westerly weakened, and we are seeing air masses coming from the Pacific Ocean, which are relatively clean marine air. So this contributes to the lower GM concentration. So overall, the large-scale atmospheric circulations plays an important role in modulating the atmospheric mercury interannual variability here. So thank you very much. This is the end of my presentation, and I will be happy to take your questions.
Thanks, Guirong. Guirong, this is Dave. I have a question for you. Do you know if the Cape Hato site is showing a similar pattern between 2014 and 17 for GEM?
You mean the capital Okinawa site?
Sorry?
You mean, were you asking the capital site?
Yes.
I don't know the detail, but I did read their paper published in Atmosphere. So in general, their data is also showing a decreasing trend of mercury concentration in marine boundary air. But I am not sure about the interannual variability at that site.
Okay, thanks.
Yeah.
All right, well, thanks for joining us, Guirong.
Okay,
Get some
thank
sleep.
you. Yeah, that's my plan.
Okay, well, so we're heading back to the US, to the west, with Peter Weiss from UC Santa Cruz. Peter, are you there?
Yes. Hi, everyone. You can hear me? I hear my echo.
Yep, you're good.
Okay, great. All right, so full screen. So yeah, thanks for having me. And the title of my talk is Investigating the Effect of Precipitation on Airborne Reactive Mercury Washout at Storm Peak Laboratory, Colorado, and at 16 co-located sites in the AMNet and MDN networks. I'd like to acknowledge my co-authors, Seth Lyman, Lynn Gratz, Tyler Elgiard, Nicole Chama and May Gustin. And also special thanks to folks at NADP, which helped me assemble the precipitation data that I used in this analysis. So the overarching question that we're trying to get at here is, can the degree of washout of oxidized forms of mercury in the atmosphere be used to provide guidance about the magnitude of low bias of KCl denuder measurement methods for Mercury 2 in the AMNet data? And so how we approached this was to inspect the Utah State University dual channel ambient mercury system, which I'll abbreviate DCS, Mercury 2 and Mercury 0 measurements at Storm Peak Lab that occurred over 2 10-month periods in 2021 and in 2022 for patterns and behavior during precipitation events. The dual channel system has been shown to be free of low bias that has affected KCl denuder measurements that have been used in the AMNet network. And then inspect the GOM, PBM, and Mercury 0 measurement data from 16 co-located AMNet and MDN sites for similar patterns. And then develop estimates of correction factors for KCl denuder derived HG2 measurements based on the comparison of washout behavior between the AMNet and the SPL data sets. Okay, so here's the study area. You can see the AMNet sites. They're co-located with MDN sites. So there's weekly precipitation composite samples analyzed for mercury concentration, and there's hourly precipitation data, and there's also the hourly or 3-hour average GOM, PBM, and HG0 data. And then the Storm Peak Lab, which is at 10,000 feet in the Rockies, collected hourly Mercury 2 and Mercury 0 data over the 2-year period, as I mentioned. There was no precipitation measurements there, but the nearest MDN site is Colorado 97, which is 9 kilometers away. These other sites are RAWS sites, which have meteorological information that was also utilized. Okay, so of all the AMNet sites, just kind of wanted to group them into general classes. And when you look at the seasonal cycle for the data at these sites, the average mean concentrations by month for GOM, PBM, and HG0, you can see different patterns popping out. For GOM, it's generally elevated in the spring. For PBM in the middle panel there, it's generally elevated in the winter. And for GEM, it's also generally elevated in the winter. And you can see certain sites pop out as having high concentrations. Others are more background, low levels. And these have been categorized. The red are more affected by local emissions. The blue are sites more in the north that may or may not be affected by local emissions, and the green is sites more in the southern US, and the 2 sites, Vermont 99 and Nova Scotia 01, seem to be the furthest removed from local sources. If we look at the diel patterns, we can draw some certain inferences as well. GOM is definitely a daytime species. I mean, there is present overnight, but it's enhanced during the day. Seems to be produced from photochemical reactions involving GEM. PBM is more elevated in the morning and the midday. The morning peak is especially seen in some of these more polluted sites where it appears that the breakup of the nocturnal boundary layer is going to be, will show the end of the peak of the PBM, so it's accumulating in that nocturnal boundary layer. And GEM is elevated at night for the polluted sites, probably boundary layer issue as well, and then mixing out during the day. But for more of the cleaner sites, it's low overnight and then increases during the day. Okay, so takeaway from all this is that despite the KCl denuder measurements being biased low, there's still a lot of important information to be gleaned from these data, and it appears that they're revealing some actual processes that are occurring in the atmosphere, despite the low bias. Okay, so now looking at precipitation and its effects on the air concentration of the oxidized mercury species. And this is something where we don't have a ton of solid information. There's a few papers out there. One of them by Zhu et al. In 2018 noted that Mercury 2 scavenging processes occur mainly in clouds and would be affected by mercury that is entrained into the clouds. So if that's the case, if there's rather ineffective mercury scavenging in the lower atmosphere and where you have your actual mercury measurement device right here at the surface, a direct relationship between Mercury 2 and air at a surface station and mercury concentration in wet deposition is likely to be weak. Okay, but despite that, if you plot up the data, and this is a plot of the mean weekly Mercury 2 concentration at the AMNET sites, and where Mercury 2 is just going to be GOM plus PBM. Some people call it reactive mercury. And on the x-axis is the median weekly mercury concentration in wet deposition from the MDN site. You can see the interesting pattern emerges where you've got these background sites with very low in both metrics and going up, and this dashed line is just hand drawn. There's no fit here, but it just implies a slope where the more polluted sites are extending up on the slope, higher Mercury 2 concentrations in air and higher mercury wet deposition concentrations. Okay, and then there's some anomalies where these suburban south sites, we know that they have elevated mercury concentration in wet deposition, maybe due to deep convection, but they're showing very low Mercury 2 concentrations in air. Now, if we look at the actual weekly means for Mercury 2 concentration, this is log of those, and the weekly mercury concentrations in wet deposition, and make a plot, and I've color-coded those by each region, we can see for the suburban north region, the suburban south region, and the rural north region, there is no relationship between Mercury 2 concentrations in the air and mercury concentration in wet deposition. Okay, so that's kind of going back to our model. It's sort of what we would expect. There's limited connectivity between these two metrics, and partly because of the timescale difference of the measurement, but perhaps also just where the mercury is coming from as it's being scavenged into the rainfall. However, the local emission sites do show a significant relationship, and one could hypothesize that this is because the rain is picking up Mercury 2 that's accumulated in the atmosphere from more of these local sources, where the below cloud scavenging becomes a little bit more important. Okay, so the 6 sites identified as being impacted by local emissions are likely contributing to local wet deposition of mercury, just based on this relationship. And then that begs the question, are we underestimating the contribution of local sources due to the Mercury 2 measurements if they're biased low? So will this slope be even steeper if we adjust the Mercury 2 data to more accurate levels? And so how do you do that? Well, that's obviously a very big question. And this is just taking one approach to see if it can come to fruition. And so what we did was look at the mercury concentration in air data, the Mercury 2 and Mercury 0, at Storm Peak Labs using the dual channel system. And this, I apologize for the units, there are nanograms per cubic meter over here. So multiply these by 1,000 to get picograms per cubic meter. You can see the concentrations measured with the dual channel system are many orders of magnitude larger than what you see with the Techran systems. And one thing we noticed, where the lines are the concentration when it was raining and the red dots are the concentration when it wasn't raining, you can see a depletion of Mercury 2 concentrations during periods of precipitation. And we also noted that concentrations of Mercury 0 were consistently enhanced during rain events, which was interesting. So looking at this data aggregated across the 2 years, segregating it based on whether it was raining according to the precipitation data from the nearest MDN site at Colorado 97, you can see that there is a consistent depletion of Mercury 2 concentrations when it was raining. And here's all the data. Mercury 2 at Storm Peak Lab was 24.6 picograms per cubic meter lower during precipitation, and Mercury 0 was 0.05 nanograms per cubic meter higher during precipitation. Okay, and these differences are statistically significant. Okay, and so we wanted to know if this was having an effect on the mercury concentrations in precipitation at Colorado 97. First of all, we looked at the relationship between the mercury concentrations in the precipitation from Colorado 97 versus the sample volume, because there is a known negative relationship. The higher the sample volume, generally the lower the mercury concentration. And in fact, we do see that, although it is a rather weak relationship, just barely significant. If we look at then the mercury 2 concentration taken over the same weekly time period as the MDN sample at Colorado 97, we see no relationship. Okay, so this is, I guess, maybe not surprising given what we know, that the mercury scavenging is coming from in the cloud and presumably much higher up in the atmosphere than your measurement station is. And in the lack of any local sources of Mercury 2 to the Storm Peak Lab environment, we might expect a very weak or no relationship. Where we do see a relationship with mercury concentration and precipitation, though, is with relative humidity. So this is selecting the relative humidity values during the periods when it was precipitating and 12 hours before the onset of precipitation to create an average relative humidity value that corresponded with each weekly precipitation sample. So it's a bit of fuzzy math, but it works out, and you can see that there is a significant negative trend. If we do the same for ozone, you can see that there is a positive significant trend. So we can surmise by this that mercury concentrations in the precipitation are higher when the precip events were drier and higher in ozone, which indicates to me that the free troposphere is an important source of mercury scavenging and ultimate delivery of mercury to the site at Colorado 97, which is consistent with the literature and the models, which indicate, especially in the Western US, that the main source of mercury and precipitation is going to be the free troposphere, the upper atmosphere, and not the surface. Okay, so given that we saw a decrease in Mercury 2 concentrations at Storm Peak Lab using the dual channel system, and you can see that by the blue bars here. So this is just showing the data that was already presented. The no precipitation mean of Mercury 2 at Storm Peak Lab was about 95, and the precipitation mean at Storm Peak Lab was about 70. And so the difference between those two is shown in the right-hand plot here as around 25 or 24.6. Okay, so then we looked at the same pattern for the AMNet data. So this is where I was really appreciative of the hourly precipitation data, because I was able to segregate the data from all these sites using a script. Of course, there's a lot of data, it would be impossible to do by hand. And for both GOM and PBM, and the height of the bar here represents the GOM plus PBM, and then they're color coded by each species. And so you can see, if we just look at the difference plot over here, that most of the sites show what I'm calling a washout effect. The loss of Mercury 2 during times of precipitation is much lower than what we see at Storm Peak Lab. And the degree to which it is lower, I'm suggesting, could be an estimate of a correction factor for the KCl denudor data. Okay, so let's go ahead and look at these correction factors by the region, the regional average for these correction factors. Now, one thing that I didn't show a plot of was the HG0 enhancement, which is probably a different process, but because it was such a stark pattern along with the HG2 washout, it's worth noting. So at every site when it was raining, Mercury 0 was enhanced at every single AMNet site and at Storm Peak Lab. And these levels are pretty comparable. The average over 2 years for Mercury 0 enhancement when it was raining or precipitating was 52 picograms per meter, or 0.052 nanograms per cubic meter. And the average of all the AMNet sites was 56. So it seems to be a pretty consistent and robust phenomenon that we are observing, and the two systems, the dual channel system and the KCl denuder system, are quantifying Hg0. Of course, KCl denuder doesn't affect Hg0. They're quantifying it approximately the same. But when you look at the Mercury 2 washout, you see a grave difference, a very distinct difference. 24.6 at Storm Peak Lab, and average vol AMNET is only 5.9. Okay, and you look at some of the regional averages, and you can see that southern region has an average washout amount of only 2. Very, very low. Okay, and so if we take the ratio of the washout amount at Storm Peak with the washout amount at all the other sites, we can get a first cut at these correction factors. And you can see they vary. Regional average for the 6 polluted sites is approximately 3. For the little less polluted sites, 5. And for the southern sites, 16. And for the more remote sites, 7. Okay, so this has some basis in the literature to do something like this. My approach is different than what was done by Cheng and Zhang, but we came up with somewhat similar results, so it's worth noting. They estimated a GOM bias factor. Their multi-year average GOM in air measurements that they looked at were estimated to be biased low by more than a factor of 2 at 6 sites. And you can sort of pick out which sites based on this plot. Some of the 2 notable ones are the GA40 and the OK99. And you can see on my table, GA40 has, well, 6.8, OK99 is the highest one that I see. But both of those belong in this southern suburban region that I defined. And it would suggest that perhaps the high humidity at those sites has caused passivation of the KCl denuder, and they're reflected in a larger bias. Okay, so I'll just wrap it up. Mercury 2 was washed out and Hg0 was enhanced in precipitation events at Storm Peak Lab and at all 16 AMNET sites. And this is looking over many years of data, and it's pretty consistent effect. It was somewhat surprising that it was so consistent, but I think that reveals some real process in the atmosphere that the KCl denuder method is revealing, along with the dual channel system. The 2-year mean Mercury 2 washout value at Storm Peak Lab measured with a dual channel system was 24.6 picogram per cubic meter. The Mercury 2 mean washout at AMNET sites was 5.9, and it ranged from 0.9 to 18.3 picograms per cubic meter, representing a factor of 4 lower than what was observed at Storm Peak. And using relative washout magnitudes, the Mercury 2 correction factors were estimated to be 1.3 to 36, depending on the AMNET site. And I think that one of the motivations that I could see was for correcting the past measurements is that it will allow for more accurate estimation of local emission sources on wet deposition at a given site and region, and the possibility that we might be underestimating the contribution of these local sources on wet deposition at some of these critical sites. And that's the last slide. So really appreciate this opportunity. I'm happy to take any questions.
Thanks, Peter. There's a question back here. Trent?
Yeah, great talk. I was just wondering if you could comment on the mechanism that you think for the enhancement of Hg0. Just how and why is that happening?
Yeah, we've talked about that some in our group meetings, and we know that soils emit mercury after rain, and Mercury 0. So that could be one possibility. There could be other possibilities where Mercury 2 is being reduced in the aqueous phase in the precipitation as it's in the atmosphere and releasing Mercury 0 back into the air. But I have a feeling it's probably emissions from the surface. But it's certainly an interesting phenomenon, and I think there's some richness that can be learned from it.
Other questions for Peter? Okay, thanks, Peter. Nice talk.
Thank you.
Our final talk in the session is from Ashley Smith, USGS, New York State Water Science Center, PFAS.
Hello, everyone. Can you hear me?
Yes.
Right, so hello, everyone. My name is Ashley Smith-Krudner, and I will be talking about per- and polyfluoroalkyl substances, also known as PFAS, in through fall at the Bronx Botanical Gardens. So PFAS are a class of anthropogenic chemical compounds that are emitted to the environment from numerous sources. They are slow to break down, and these characteristics lead to their accumulation in the terrestrial and aquatic ecosystems, posing a possible threat to human and ecological health. PFAS may become airborne, allowing them to be transported long distances through the atmosphere before being removed by wet and dry deposition. And measurements of PFAS flux and deposition rates, however, remain sparse and underdeveloped. So in 2020, the NADP had conducted a 5-month study that documented PFAS concentrations and deposition of fluxes and precipitation across Wisconsin. Samples were collected from 8 national trend network sites, and those samples were analyzed for 34 PFAS compounds. Concentrations range from 0.7 to 6.1 nanograms per liter, with a median of 1.5 nanograms per liter. Analysis of regional PFAS compound profiles had indicated possible source influences. And this study kind of provided some guidance in our study in terms of how we sampled and we'll later use our data. So in 2019, the USGS had conducted a surface water monitoring study that documented the presence of PFAS within the Bronx River. Analysis of data from 6 different locations during both dry and wet weather showed a consistent PFAS mixture and concentration. These results, along with the industrial and highly urbanized setting of the Bronx River, suggests that PFAS occurrence is influenced by atmospheric deposition of PFAS in this area. The image to the right is showing an overview of a section of the Bronx River that's flowing through the Bronx Botanical Gardens, which is also the location of our sample site. Not much is known about the magnitude of atmospheric PFAS contamination on the Bronx River or the surrounding area, and that brings me to the objectives of our study. So this study aims to identify the magnitude and mixture of atmospheric PFAS in through fall, litter fall, and the surrounding landscape near the Bronx Botanical Garden, or in the Bronx Botanical Garden. It also aims to document the magnitude of atmospheric PFAS in the urban environment that could potentially pose risk to human and ecological health. The pictures on this slide are just showing the front of New York Botanical Gardens, or in the Bronx, or Bronx Botanical Gardens, and this is an image of the Bronx River at the gardens. So our study site, referred to as 06NY, is indicated by this yellow star on the map. And this image at the bottom of the slide is showing an actual view of our site. Also at the Bronx Botanical Gardens is the NADP site, NTN NY06. where open air precipitation samples are collected. Let's go next. So our study site is specifically at the Thane Family Forest section of the Bronx Botanical Garden. The Thane Family Forest is the largest uncut expanse of New York's original wooded landscape. And our approach to the study is mimicking a current NADP study that's taking place in Duke Forest, North Carolina. So as a part of our approach, we're collecting weekly precipitation samples throughout the growing season using through fall collectors. So this collection time period is going to be from April 2023 to November 2023. And our samples are going to be analyzed for PFAS, general chemistry, and DOC. Results are going to be compared to open air precipitation samples that of the open air precipitation samples, which are being collected at the nearby NY06 site. From September to November, we're also planning to collect litter fall samples, and those samples will be analyzed for PFAS and mercury. So on the slide, you'll just see a picture of one of our through fall setups. Here's the through fall collector. Each setup for all of our through fall collectors look the same. They're all labeled, and they're all just zip tied to the pole and secured that way. This picture just shows the HDPE mesh, so the top of the through fall collector. And here's just a group of the multiple collectors that we deployed out to the field. So the results of this study will help fill in the knowledge gap in the identification, transformation, magnitude, and exposure of atmospheric PFAS within an urban environment that could potentially pose risk to human and ecological health. Right. So at our sample site, we have 7 through fall sampler collectors deployed. They are retrieved and deployed weekly on Tuesdays, aligning our collection days with that of the open air precipitation sample collection that is taking place at NY06. Sample bottles are weighed before deployment and after retrieval. And depending on the volume of sample collected, the 7 samples are either composited and analyzed, or each of the 7 samples are analyzed separately to offer insight into spatial variance in PFAS mixtures and concentrations. As said before, the collection is being done from the beginning, starting from the beginning of the growth season, so throughout the growth season, so from April to November. And next. All right. So as mentioned earlier, litter fall collection began in September and will be ending in November or early December. 4 litter fall collectors have been deployed in the same area as the through fall collectors. Litter fall samples are collected every 4 weeks and will be analyzed, as I said before, will be analyzed for the 34 PFAS compounds and mercury. The methods that we are using for litter fall collection are following the NADP Mercury Litter Fall Network Site Operator Guidance. So as of today, we only have 2 weeks of PFAS data available. They're all preliminary. This graph represents data from through fall in green and open air precipitation in red collected in week 1. During week 1, total PFAS was substantially higher than in open air precipitation. 26 of 34 PFAS compounds were detected in our through fall samples, while only 6 of 34 were detected in the open air precipitation samples collected from site NY06. And this is pretty much along the lines of what we expected to see at the sites. During week 2, only 1 PFAS compound was detected in open air precipitation, while 20 out of 34 of the PFAS compounds were detected in through fall. The one compound that was detected in the open air precipitation was found at a greater concentration than in the through fall samples. Right. So based upon our preliminary data, we find that PFAS is present both in open air precipitation and through fall. The PFAS concentration measured in through fall are much greater than in open air precipitation. And lastly, several PFAS compounds that were detected in through fall were not detected in the open air precipitation. So the next steps for our study include continuing collection of both the through fall and the later fall samples until the end of November or early December. And following completion of sample collection, we plan to compare study results to that of the open air precipitation at site NY06. Great, well, thanks for listening, and I welcome any questions. And
Thanks, Ashley. Nice talk.
Thank you very much. That was a great talk. I noticed that PFBA is the most common analyte that you found, either in through-fall or precipitation. And I'm wondering if you looked at any of the carbon count of all the PFAS we're looking at. I think PFBA is a C4, so that's probably one of the smallest. That's the question, sorry.
So I just recently got this data, so I haven't been able to kind of look at, the broader picture as of yet. But if you want to contact me by email, I can definitely get those answers for you at a later date.
Sure, thanks so much.
No problem.
Mike?
Thanks for this. It's really exciting to see both. I mean, it's terrible to see this through fall enhancement of PFAS deposition, but I'm really excited to see you bring in the ecosystem component in, and I'm really excited. I want to see the litter fall results. But I was just thinking, like, in taking that a step further, we were talking earlier about like wildlife impacts, and if you have any plans or you know of anyone like looking at potential data from herbivores in the area, like whether it be hair samples or muscle tissue samples of like looking at PFAS accumulation in local wildlife.
This project was only for, I guess, 2023. So after this, we're done with this project, or USGS is done with this project. As for just studying any tissues or any animals or hair or anything like that, I do not know of any studies that are focusing on that right now. But maybe that's the next step. Maybe the USGS can jump in and do that. But it's a really great question, something to think about.
So I can address that too, Mike. The environmental health program with USGS just is wrapping up a study looking at deer meat and other like hunter harvested meats from around the country. And I think that probably the preliminary findings will be coming out in the next 6 months or so, but I can put you in contact with the leads if you're interested. Yeah. Okay.
Okay, I think we thank you for your talk. Thanks, Ashley.
Thank you.
And this concludes session 3, and let's thank all the speakers for the really excellent talks today.